amateur. I've had a few requests recently to go a little bit more in depth into my interview like kind of experience and what kind of questions I was asked, what you can kind of include in your personal statement. I am going to do a full video on personal statements very soon, but for now I'm just going to kind of talk about the interview process, but more kind of like of an academic viewpoint rather than just a social and how it made me feel. So without further ado, let's get on into it. If you've watched my interview video, when it's like clickbait interview 3 hours sleep, it's not clickbait, I actually did it on 3 hours sleep, but I went down after my 18th birthday party and I was hungover. I'd had 3 hours sleep, I was very tired, and they said, okay, at 2 o'clock today. I arrived there at about 1 o'clock, and this was like, okay, at 2 o'clock they said, right, you've got about an hour, you're gonna be the third person that's interviewed, and good luck. I'd anticipated lots of coldness, lots of snide comments or things like that, and actually there wasn't any of it really. I didn't feel out of place, people weren't snobby, people weren't nasty or anything, so it was a really pleasant surprise. I got called in for my interview and the first part was half an hour. I was timed by a second year student who was in the room with me. I was given an article to read and some highlighters, and they said, okay, we want you to highlight the, I can't remember whether it was 3 or 5, I think it was the 3 most important things that I read in the article. So I had a pen and I could go around and write bits and pieces on the notes or whatever, and then I had to go through and highlight the 3 most important pieces of information, and it was actually on something called GFP, which is the green fluorescent protein, and essentially it's what we can, when you see how scientists have made mice glow in the dark or whatever, this is the GFP protein, so we use it as a marker to kind of show that things, genes that we want to insert have been inserted. I highlighted lots of different things. I can't remember exactly what they were, but they were kind of like along the lines of their use for identifying when other genes are inserted, and about the structure. I did a point about the structure of it, and one that it was actually glows in the dark, because otherwise if it didn't glow in the dark it wouldn't be of use to us, because we wouldn't be able to tell that that gene has been taken up by a cell or an organism and whatever. After my 30 minutes reading, they took me up, another second year student kind of ferried me from where I was reading up into the academic office, and so then I was just kind of didn't know what to expect, didn't know who I was going to have. I mean, I'd met them both, I'd met luckily the 2 tutors that interviewed me before that, but I didn't know who was actually going to be interviewing me, whether it was going to be one of them, whether it was going to be both of them, I didn't know. I was really surprised. I went in and there was just paper everywhere, books, the fire was on, they were just sitting on armchairs, really casual. It was like being in someone's living room where like a paper bomb had gone off. There was just bits and pieces everywhere, but it was really homely and really warm. Both of the tutors at Hertford were really accommodating, really friendly, didn't make me feel on edge, just wanted to chat to me, and it really felt like they were just trying to get a feel for what I'm interested in and what I think about certain things and whether I could do things, and you know, it was a real pleasant surprise. It was not by any means someone sitting there going, what's the answer to this, what's the answer to that. It is a discussion with people that can actually really enrich your knowledge, and it gives you an opportunity to talk to some of the top academics about your opinions on things, and I just don't think there is anything more exciting than that. So the first thing that we did was we sat down and we looked at a photograph of a worm kind of thing. And so basically I had gone on a summer school and had learned a bit about nematodes, which are really small, probably like a millimetre I don't even know. They're really, really small. You can barely see them with the naked eye. And they're like basically tiny, tiny worms that you can genetically engineer, and because they reproduce quite quickly and things like that, it's good for like testing genetic engineering on a lab level. And so they'd done a few kind of examples with us, with that actually with GFP. So I luckily had recognised them and I knew what they were showing me, and that's the first question they asked. It was like, what would you think this is? You know, now if you were in that position and you didn't know what they were, you would guess. And you know, there were parts in the interview where I had to guess things. You know, they would ask me questions about things that I didn't know about, and you have to make an educated guess. You have to think, well, it looks like this, so I'm going to say that, and they will correct you. They will say, not quite, or well, what if I told you if it was this, and then go on to ask you something else. And using the information that they feed to you, you just have to keep on going. So then they started feeding me kind of like bits and pieces of information. We sat down and they just showed me a sheet with these different types of nematode on. So one was just like what I knew to be a normal recognisable nematode, and they asked me, oh, what's this? So I told them. And then they go on and say, okay, well here the nematode has a gene that causes the nematode to be slightly shorter and fatter and a little bit more curved, and then this one is like longer and thinner, and this is to do with genetics and whatever. And so they started probing my knowledge of, say, dominant and recessive alleles. They started prompting me to link genotype and phenotype together and talk about different bits and pieces. And so then they gave me a sheet with different combinations of alleles. So they told me that there was an allele for short, fat and curved. They told me that there was an allele for thin and long, and I think one of them was that it was sterile, or it couldn't reproduce. I can't remember exactly, but I know that they were about different kind of genotype, phenotype. And from those alleles then I had to deduce which ones were recessive, which ones were dominant, and so etc, etc. And you just had to do that using the information that they'd given you, and they'd given me enough information. They're not going to expect you to just know these things off the top of your head. They're going to give you bits and pieces of information and expect you to get there on your own after they've given a little bit of prompting, a little bit of information, and make an educated guess even if you aren't sure. I wasn't sure. I just said that I was just talking through everything that I was thinking. So if I had a thought, so I said, okay, well, if that is thin but its genotype has this allele and this allele, then actually it must be the thin allele that is dominant because that's shown in the phenotype. And I would work through all of those kind of thoughts that I was going through in my head aloud, so that they knew exactly how I was coming to my answer, so that even if I was wrong, they'd still understand why I said it. If you just go in and explain why you think it in the first place, that's going to come across as much more confident, much more comfortable with your knowledge and your skills, as someone who just goes guess with no kind of background or as to why you think it. Then they moved on to the article. If you do have pre-reading, don't be surprised if it's the first thing that you go in is not the thing that you read about, because I think that they do it on purpose so that it's more on, you know, on your feet. So they would say, okay, why have you highlighted the fact that you think it's green and fluorescent? And it was almost as if they were saying, well, yeah, it's green and fluorescent, why would you put that on there? Then I was going like, well, if it was never fluorescent, then you wouldn't be able to use it as a marker because it wouldn't show in the phenotype. So the protein produced has to be green and fluorescent. That is a key thing in the article, because otherwise all the uses that it goes on to talk about wouldn't be true. And then we kind of moved on. We talked about why genetic engineering has cons and has benefits, what you would use it for, things like that. All things that I won't go through now because you can research them on your own. They think about things, especially if it's genetics or proteomics, anything like that. You know, make sure you're reading up on pros, cons, practical techniques, things like that. You know, gel electrophoresis came up many times. I know that some people learn about that at A level, some people don't, but I read about it in much greater detail and actually had the opportunity of doing it. And so that was really, really helpful because I was much more comfortable with what they were talking about. They gave you enough information for if you'd never seen any kind of gel electrophoresis or anything like that, then you would have been okay, but it's a good thing to research because it might make you feel a little bit more comfortable about the things that they're asking you. So after we discussed the article, they just kind of asked me a few general questions about genetics, and about DNA replication and the order of things that happen, and that was about it. They said, okay, thank you very much for letting me speak to you, and that was my interview at Harvard. Then we get on to my interview at Queen's. It wasn't awful, but it made me feel much more on edge. Maybe that's because of the surroundings being new, because the tutors being completely new. That kind of threw me off when I was waiting. So I was outside and it all felt very foreboding. It was night time and really big, prestigious, historical college that was like so totally symmetrical and everything like that, and it was really kind of off-putting. I felt really kind of on edge. But I was waiting outside and I just thought, you know what, like, what is the worst that can happen? They say no. That is the worst that can happen. And if that's the worst that can happen, why not just go in and enjoy it? That's what I told myself outside, and I kind of pepped myself up and I thought, you know what, I can do this. I went in and I was feeling really good and I tried to make them laugh and nothing, you know, no smile, no nothing. And then I was getting more and more nervous. So I got a B in AS Maths and they said, okay, so we're going to test your mathematics skills. And that made me nervous as well because I thought, okay, is this because they think it's a weakness of mine? Are they going to give me some really mad, crazy, hard equation that I'm going to have to solve in front of them and I'm going to be like shaking with the pen? But actually, it was okay. It was a couple of indices type questions, a couple of logarithms, and nothing that you would see beyond like starter questions in maths. And then they moved on to rate equations in chemistry. So I had a complete mind blank and they were asking me about things and I literally just turned around and I was completely honest. I said, look, at the moment all I can remember about rate equations is that you add the powers and that's the order of the reaction. And they were like, okay. So then they started prompting me and giving me bits and pieces and saying, okay, well, could you draw what maybe this graph looked like or what this rate of reaction graph would look like? It was okay, and in the end I remembered a lot more. It was just the nerves that completely overcome me, and I thought, this is a topic that I should know about. Why can't I just do it, you know? I made a mistake, and so will you probably in the heat of the moment, under all of the nerves and all of the pressure that you put on yourself. You will make a mistake, but that is not to say that you are going to come out and not get an offer or to not get into a great university or whatever. Like, I made a mistake and I'm going there, you know, they didn't say no, it wasn't an issue, and I got into my first choice college. So after that they talked to me a little bit about proteins and how genetic code and proteins are linked, i.e. order of amino acids corresponds to the order of bases and things like that. And so it was a little bit of maths. If you don't know, then you can use gel electrophoresis on proteins, not just on DNA. You have to use a smaller webbing gel, and you have to stain it with something called SDS, which essentially gives it a negative charge so it moves and it makes it more kind of stick-like. I don't know, you know, it just makes it breaks the bonds kind of thing, so that we're just dealing with size rather than shape and all of that malarkey. The specific premise that I was given was a scan of a gel electrophoresis sheet, I don't know what they call them, but where you can see all the bands. If this protein was inhibited, what would it look like on a gel electrophoresis sheet? And now I'd have to draw the bands on myself as to whether if something was inhibited there would be more of a protein or less of a protein. Now obviously I can't I'm not going to go into the ins and outs of why I put answers that I did or why I did this. It's just to make you aware of the kinds of things that they're going to probably ask you to do. The circumstance that you might be asked things in will be different to what it was for me. So they're not going to ask you the same question, they're not going to ask you to do, they're not going to give you the same sheet with all the nematodes on or the same gel electrophoresis sheet. They are going to ask you something new, but with the same kinds of ideals. So the specifics aren't important. Getting a general idea of practical techniques, getting a general idea of beyond your A-level specification, things like proteomics and how labs measure the size or the charge or whatever of proteins, and all of those things are going to help you be more comfortable with what they're asking you in the interview. So the aim of the game is to make sure that you are familiar with as many things as you possibly can. Like, go over your chemistry spec, especially things like rate equations, especially things like thermodynamics, and all of that is going to make it much easier for you to answer quickly and concisely, and so that's the main thing. I've told you the kinds of things that they ask about, but obviously each person's interview is going to be slightly different, and so you just have to practice using those skills and thinking on your feet. Your chemistry teachers, your biology teachers, these are the best kinds of people to get on board and ask you questions. Get some of these kinds of questions and get your parents to ask you them as well. On top of that, although your parents might know nothing about biochemistry, get them to ask a question just so you can practice answering. As long as you're used to answering questions about certain topics in biology or in chemistry and giving sufficient detail, that's a great skill to start with. So if you do that, then you can move on to having like practice interview questions with chemistry teachers, biology teachers, maths teachers, who knows. So yeah, that's about it from me. I hope to see you soon. I will definitely put up a video about personal statements. If you've got any questions, put them down below. Until then, bye bye, see you later.